Great pleasure to welcome today's speaker, Alexander Rauch. Alex is a senior postdoc in my lab, and he's been working with me since 2012 now, and he will soon be heading out to start his own group, hopefully in the context of SDU. That's what we're aiming for at the moment. So Alex did his undergraduate studies at Jena at the Finnish Schiller University, and then he moved on to do his PhD in the Leibniz Institute of Age Research with Jan Tuckerman. And I got to know Alex when he was a PhD student. I was lucky to meet him at a summer school in Spetses in Greece. And again, maybe a year later or so, in a meeting in the US, I managed to convince him that SDU was a great place to do a postdoc. And so we worked on applications together, and Alex obtained an EMBO long-term fellowship, came to SDU and started, as I said, in 2012. So it has been an immense pleasure to work with him, both scientifically as well as a personal level. And I really hope we can keep this interaction going even when he flies off as an independent researcher not too long from now. So with that, please.
Thank you, Susanne. Maybe I should just mention to that before I came here to Denmark, we met not only on 1 or 2 meetings, I think it was more like 5 or 6. Oh, we had a really intense conference relationship, I would say, during this period. Today I would like to give a presentation about the work which I have been involved in more or less the last 5 years here at SDU. And I want to bring it into a bigger context of asking if fat cell or becoming a fat cell is actually the easiest choice the cell can make. So here we have an electron microscopy picture of a fat tissue. It looks very, very nice. So this is done by Steve Schmeissner. He is an artist, I would say, who has access to an electron microscope, and he's making a lot of very, very beautiful pictures can be found on this website if people would like to use these kind of pictures for their presentations, just mentioning. So today I want to talk a little bit about fat tissue and fat cells. And I guess everybody of you would agree that the fat tissue is more or less very important to store energy. Especially this attribute of storing energy has been challenged of the way how we think is as a society, as nutrients are more or less quietly accessible these days. And fat tissue is not only important actually to store energy, but it's actually also involved in regulating appetite or lipid metabolism. It's important for insulation, but also padding, so with respect to the skeleton. And it also actually acts as an endocrine organ because it can regulate things like blood pressure and also glucose levels. Fat tissue is more or less like all the other tissues in the organ also subjected to a process called aging. So when we just look, for example, here at the fat tissue, which is underneath the skin in the face, and comparing a young face compared to an old face, you can see that the yellow stuff, which are the fat tissues, they shrink actually during aging and also contribute to, in this respect, to all the wrinkles we develop during aging. But I don't want to focus so much on places where you usually would suspect fat cells to be. So like in the fat tissue, I actually want to focus more on organs and places where you usually don't think about fat cells in the first place. So here we also have, for example, muscle tissue from an individual, a more younger individual and an older individual on the right side. They were matched based on the body mass index, so they are equally obese, so to say. However, you can see that during aging, you lose quite a lot of muscle mass, while at the same time the intramuscular fat content, which is more or less the white stuff here within the muscle, actually increases. So you get more or less a redistribution of fat during aging. This is not something which is special to humans, so this is also something you can find in other organisms, much simpler organisms, like for example C. elegans, which is a nematode. And it's just showing here at this worm at day 1 and day 9. And what we are looking at, the fat is actually stained with this blue dye. So could we maybe turn the light a little bit? down. Yes, so then it's easier to see in these fluorescent pictures that actually the fat content here also in these, say, elegans or nematodes increase during aging, and also similar in the fruit fly. So this is also a very well studied model organism, and this is actually particular, the region where it actually fat accumulates is in the so-called jumping muscle, which is located here in the organism. It's shown here by this blue staining down here, and what you should just emphasize is at this panel you have increasing more green dots coming up, which should indicate lipid deposition. So there's also accumulation of lipid during aging of Drosophila going on. Interestingly, this, as I said, is not only taking place in tissues that would expect fat, or maybe in metabolic tissues, but it actually also takes place in tissues like the thymus, which is very important for our immune response. The thymus is actually the organ where our T-cell precursors, which are derived from the bone marrow, they enter the thymus and then have to go through maturation to more or less produce mature T-cells, which are characterized by a T-cell receptor. This is very important for antigen recognition, so if you have microbes invading you, they will recognize it by the T-cell receptor. And meaning the word naive T-cells means just that they have not met their antigen yet outside of the thymus, and they are selected in the thymus actually to not respond to our own proteins. When you then look at the thymus, what the structure looks like in a 4-year-old human individual compared to a 40-year-old individual, you can see that the so-called cortex and medulla, so this is the area stained with this purple dye, actually shrinks tremendously during aging. These are the places where the T cells mature, and we have a lot of increase of the white stuff, which is actually fat. This is also shown down here, more or less a graphical illustration that by the age of 20, actually there's already a dramatic decrease in the functional tissue volume of the thymus, and it's just replaced by fat tissue. So in other words, if people or students are coming to you, at least from my point of view, and asking about your age, you can almost say that depending on the thymus, we are not much different anymore. Yes, so despite the thymus, there's also another organ. It's an organ where I'm interested to look at, which is bone. And bone is, for most of the people, actually just a very static kind of tissue. Maybe the only, more or less, experience you have with bone that it hurts tremendously if you break it. But other than that, bone tissue is also a very, very dynamic organ, so to say. And I would just to invite you to take a tour in this video, which should just show how actually bone biology works. So bone is, of course, part of our skeleton. It gives us our structure, what we look like, very important to discriminate us from jellyfish. And bone has a very intense anatomical structure inside. So usually you have this called cortical bone, which is very enriched for collagen and matrix, which gives the stiffness to bone, whereas inside you have the so-called trabecular bone, which is almost like a sponge, which is very important for the elasticity of the bone. So depending on how you mix these two, it's important for the how the bone can be bended or when they break. Looking a little bit inside to the bone, it's not just extracellular anorganic matrix. There are actually a lot of cells sitting on it and working on these. So there was one cell type which is called the Osteoclast. This is a cell of hematopoietic nature, so it's a blood cell-derived cell. And these cells are really sitting on the bone matrix and crawling along and removing bone. So they remove bone every day. And this is really what it looks like if you just place it on an artificial bone matrix. They crawl along and remove it. And that's how so they generate an acidic condition where they can actually remove the bone. So they would be actually very nice having you to clean your bathroom if you want to remove all the lime. There's another cell type which then has to rebuild bone. This is the so-called osteoblast. They are also sitting more or less on top of the bone matrix, illustrated here that they're crawling along and they actually build up new bone matrix. And that's most important by just producing a lot of collagen. That's the most or the biggest component of the bone, which is then calcified. So bone grows by the action of the osteoblasts. Some of these osteoblasts are not only crawling along and producing a lot of matrix, but actually what they also do, they start to build themselves into the matrix. So meaning they produce a lot of matrix, it's getting calcified, and at some point they are inside the bone matrix. And at that stage, we don't call them osteoblasts longer, we call them osteocytes. They have a very neuron-like structure, and they are all connected within the bone, and these are actually the cells sensing if you break the bone. You could imagine that these two cells, which are important to resorb the bone and build up bone, actually are not imbalanced anymore. There could be a tremendous issue for the bone mass, the resulting bone mass. And this is something what we call, for example, osteoporosis, where you have, if you have more resorption going on than building up new bone. And this is very care intensive in case of hip and spine if they break, because they're involved with costing a lot of money. And within the bone, there's not only the bone cells which form the bone, but there are also a lot of other cell types like the hematopoietic stem cells. So meaning having a good bone compartment is also important for the development or the maturation of all our blood cells. Yes. Some of you might think bone is almost like traffic work, right? So commuting along Urbeckwine might just see what bone remodeling means. So you have removal of asphalt and getting a new layer on top of it. I guess that's treated more or less every year. So bone is a little bit more dynamic, but you can relate it to these kind of comparison. The nice thing about bone is not just decreasing, so you can actually also train your bone mass just by exercise. Maybe whereas in the old days people ask, hey, do you want to lift some rocks for 50 fish? And you would say, yeah, of course I could use some extra fish. And then say, no, I'm charging you to lift the rocks. And I would say, oh, that doesn't make sense. Actually, these days people do this kind of way. They go to the gym, pay a lot of money, and actually to train the body. And with respect to bone, this is actually a really good thing. So moving actually is already very good because just sensing pressure on the bone by the osteocytes is very important to keep up bone structure. If we now look...
So it's not good to be researchers. That's the...
So when you look inside the bone then, so we're looking here at the bone marrow histology of a young individual and then older individuals, you can see that these white spots, which are actually adipocytes, increase during age, meaning that the bone marrow gets more or less fatty during aging. A side effect of that, you might realize that all the stained area, so all the purple stuff, which is usually represented here by hematopoietic cells sitting there, which are the most frequent cells in the bone marrow, actually decreases, right, due to the increase of the size of the adipocytes. So taking this into account, one could also say that actually the output of our hematopoietic stem cells, what the bone marrow produces, also decreases during aging. By the same time, the fat tissue increases. And when we relate this to what I told you earlier about the thymus, is actually that we have during aging less output of T cell precursors, while at the same time our thymus is also not working that well, both related to with the fat tissue content. And this could be give some explanations why actually older people, or more or less older people, are actually severe quite much more from the childhood diseases like measles, mumps, or these kind of diseases, where usually at the childhood can easily fight these diseases because you have a rich pool of native T cells, but these decrease during aging. So this is what it looks like when we take sections of a bone, having at the top a young bone, at the bottom an old bone. And on the what is stained here in this section with this black dye is more or less the bone matrix. So this is something giving the stiffness to the bone. Everything is black, is more or less calcified matrix. And you can see, comparing the young and the old, that there's less black staining, meaning you have less bone mass. This is the condition what we call osteoporosis. While at the same time, if we do a CT scan and look for fat tissue, which is indicated here with the yellow color, actually increases, which somehow indicates that there might be an imbalance, and one could provocatively ask if osteoporosis is just the obesity of the bone. Why ask people these questions? And this is actually due to the fact that the cells which produce the bone, the osteoblasts, which I showed you before, which put on the new asphalt, so to say, and the fat cells actually derive from the same precursor. So they have the same stem cell, adult stem cell. And the stem cells we are looking at here are the so-called mesenchymal stem cells. They are found in various organs in the body, so more or less all organs which have a certain kind of structure. They produce a lot of this called connective tissue. What we can isolate these mesenchymal stem cells by digesting more or less the tissues, putting them into a tube and into a centrifuge, spin them down. They will be at the bottom of the tube, and then we can put them onto a culture dish. And one of the nicest attributes of these stem cells is they will adhere to plastic and start growing. And then we can actually investigate these mesenchymal stem cells in the culture dish because they will still retain their potential to form osteoblasts or adipocytes in vitro within the culture dish. So to investigate how they are working. So what we started when I came here to the lab, we had access to a so-called cell line. It's a mesenchymal stem cell line which was generated here at the hospital in Odense by Mustafa Qasim's group. They were derived from the bone marrow of a healthy male donor, and they were processed a little bit in a process for biology, we call it immortalization. And this just means that we can more or less expand these cells unlimited in culture. Usually primary cells won't do this, they will stop proliferating at some point. However, these kind of cell lines, they can be expanded, so we can have a lot of material to work with. And what we did with these cells, we can put them into the culture dish, as I said, and then we have two kind of different cocktails to stimulate them either to become an osteoblast or adipocyte. So we can direct it with adding different hormones. And the only thing I want to emphasize at that point is that both cocktails actually contain dexamethasone. This is a synthetic glucocorticoid, and I just want you to keep the word glucocorticoid in mind for some time. Just put it into your memory. Glucocorticoid, glucocorticoid, glucocorticoid. Okay. So the process of differentiation looks a little bit like this. So having adipocytes and osteoblasts, so the top panel is osteoblasts and adipocytes. They undergo some morphology changes. The osteoblasts seem to get very extended, spindle-like. They more or less grow in a mesh. Whereas the adipocytes, they actually seem to increase in size and actually also accumulate lipids by the time. So this cell here has started accumulating lipids. It's not so easy to see in the phase contrast microscope, but we can stain these lipids with dyes afterwards. So there's a dye which is called OLRED-O. It specifically stains more or less lipids which are stored in the cells. And this is indicating here that the cell which has received the adipocyte cocktail forms these lipid droplets in the culture dish, indicating that it has become an adipocyte because this is what it should do, storing fat. Whereas the osteoblast produces a lot of matrix. These matrix should be calcified, and this is something which we can stain with alizarin red. So this is a dye which recognizes calcium, meaning indicating that these cells here in the culture dish also produced a lot of lime, which can be stained. So of course these cells differ in their so-called what we call phenotype. So the stem cell does not produce a calcified matrix or neither has lipid-loaded droplets. The adipocytes have, as well as osteoblasts, produce matrix. So they are different in their phenotype. But what you would agree on is that the genetic information which these cells contain is similar between the stem cells, osteoblasts, and adipocytes because these two cells are derived from the stem cell. So it's not the genetic information which is different, but it's the usage of this genetic information which differs. And this should be illustrated here where we have the DNA, so our genetic material is wrapped around histones in the cells. It's relatively inaccessible, so meaning that the DNA is more or less protected by the histones and indicated here with these arrows as the location of different genes. And some genes might be only active in the stem cell, some genes might be specifically active in the osteoblast, and some genes specifically in the adipocytes. So meaning that in the end, these cell types will produce very specific proteins related to their function. Adipocyte, like lipid storage, osteoblast, like matrix mineralization. And mesenchymal stem cells, for example, are very important for the support of hematopoietic stem cells. How can we measure gene activity? There we use a method which is called RNA sequencing. So in order to measure gene activity, we measure actually gene transcription. This is done by a protein which is called polymerase II. It's sitting on these active genes and produces a lot of RNA molecules. And these RNA molecules, or messenger RNA, is used then later to translate it into protein. So we're not looking at the proteins, we're looking at an intermediate step of the production of protein, of gene expression, which is RNA, and this can be easily isolated chemically and then can be sequenced these days. So we can get a lot of information what is going on in a certain cell. It ends up with a matrix, so it means that you have a long list with all the gene names, and then you have your conditions, so your samples, and more or less indicating numbers, how much activity of a certain gene is in that sample. Of course, we're not only interested in the endpoints, so when we have the phenotype, because differentiation is a gradual process. We also sampled time points before we actually have the phenotype at 7 days, 3 days, 1 day, and 4 hours of stimulation to really see something what is going on the time scale. And this is what the data looks like. Out of the approximately 20,000 genes we have in the human genome, we find 5,000 of them which change expression during differentiation either to osteoblasts or adipocytes. And the way I will often represent these data is that I have the stem cells allocated here in the middle, and adipocyte differentiation is always to the right in the red color, whereas osteoblast differentiation is with a blue color to the left. And what you should appreciate from this heat map, which has more or less these 5,000 different RAWs, 5,000 genes, is that a lot of genes change expression during adipocyte differentiation. They get upregulated or here downregulated. So blue means less, red means more of the RNA. Whereas during osteoblast differentiation, it seems to be not that many changes in terms of quantity compared to adipocyte differentiation. So let's just not look only at quantity, let's look at quality. So we look at some specific gene clusters, and clustering means that these genes have a certain common temporal profile during differentiation. And isolated here a cluster where genes go up during osteoblast and during adipocyte differentiation, genes specifically going up during osteoblast, or genes specifically going up during adipocyte differentiation. So I hope your memory is still working, focusing on this group of genes being upregulated during osteoblast and adipocyte differentiation are enriched for genes with respect to glucocorticoid signaling. So that's a nice control because both cells differentiation contexts receive glucocorticoids, and we can see that the cells respond to the stimuli. We can see that these genes which are upregulated during osteoblast differentiation down here are enriched for terms like bone remodeling, ossification. So which means that we already know a lot about these genes, and the genes we're measuring here, they have been related previously or functionally be annotated to these kind of terms. Similar for the genes going up during adipocyte differentiation, they have to do something with lipid storage and fatty acid metabolism. Also totally makes sense for adipocytes. If we now ask the question for these three groups of genes, how high are they already expressed in stem cell state? We can see that especially the green ones, so the ones which are usually upregulated during osteoblast differentiation, are expressed at the highest level. So the stem cell already express a lot of the genes which usually go up during osteoblast differentiation, whereas the ones upregulated during adipocyte differentiation expressed at a lower level, indicating that the osteogenesis processes involve upregulation of many genes that are already present in the stem cells, whereas adipogenesis involves the induction of genes that are inactive in the stem cells, but actually also, looking here at the top, actually also involves the downregulation of a lot of genes which are active in the stem cells as well as in the osteoblasts. Okay, then you might say, oh, you looked at the cell line which have been isolated from the bone marrow. Maybe it's not that surprising that not so much is going on during osteoblasts compared to adipocyte differentiation. In order to challenge our model system, we then use a cell line which has been isolated from the human adipose tissue. We got this from Peter Tatrai working in Hungary. We did exactly the same experiment, they got exactly the same cocktail, and just to confirm that also these cells derived from the adipose tissue can form or calcified extracellular matrix under osteogenic conditions, they form lipid droplet lipid-filled cells during adipogenic stimulation. We then did RNA sequencing and compared what's going on. On the top row, it's more or less all genes which were differentially expressed during osteoblast differentiation. The lower one is the what genes differentially expressed during adipocyte differentiation. We can see that the quantity, again, adipocyte, there are more genes regulated compared to osteoblast differentiation. And interestingly, when we compare just both heat maps, just visually, they look very alike. Also meaning that the gene profiles or the programs we have going on are very, very similar between the two cell types. You could also ask this in a different way, using a principal component analysis, more asking about the variance in your data set. This is represented in the principal component analysis. And we can see that PC1, which describes about 60% of the variance in your data set, so how much the genes differ between the samples. And this component actually separates quite nicely our adipose tissue-derived cell line compared to the bone marrow-derived cell line. So the cell lines are very different, However, if we look at PC2, which reflects 26%, it nicely shows the resemblance to adipocyte differentiation, whereas PC3 resembles osteoblast differentiation. So somehow mimicking this one, that the gene programs are very similar, despite the cell lines are very different. So from this we concluded actually that irrespective of tissue origin, adipogenesis is associated with more dramatic changes in MSC gene expression compared with osteogenesis. Okay, we can still argue, well, you're just looking at cell lines, they are immortalized, they're not really primary. We got in contact with Camilla Scheele from Copenhagen University and got stem cells derived from white adipose tissue, brown adipose tissue, as well as from muscle. And we got bone marrow derived mesenchymal stem cells from Mustafa Kasim here at the Odense hospital. We got these stem cells and differentiated them to adipocytes, osteoblasts, did RNA sequencing again, and looked what the data. I want to present you what the data looked like. So first of all, using a principal component analysis to see how much the samples differ, we can actually nicely see that by component 1 and 3 we get a separation of the tissues. So irrespective of if the cells are differentiated towards osteoblasts, blue, red, and adipocytes are not differentiated, more or less the ones coming from the muscle tissue are very close together concerning PC1 and PC3. Here we have all the cells derived from the adipose tissue, and down here we have all the cells derived from the bone marrow. So which indicates there is of course a very big difference depending on where the cells are coming from. However, if we ask, can we also see by principal component effects of differentiation? Yes, we can, and this is reflected by PC2 and PC4. It actually nicely separates the response to the adipogenic stimuli and the response to the osteogenic stimuli, and now it seems to be very irrespective of where the cells were coming from. So that's a nice way out to look that these cells can respond in a similar way to the cocktail. So also indicating, again, irrespective of tissue origin, adipogenic stimulation drives more changes than osteogenic one. The next thing is that we wanted to know a little bit more what kind of factors are actually involved in this process. So not just looking at quantifying gene expression, but also really to identify or pinpoint factors which control this differential gene expression, what we call it. And in order to estimate how transcription is controlled, there are some elements in our DNA which are called regulatory elements. So they have been described for several years. So these are actually sequences in the genome which don't code for protein, but they are very important to control the expression of RNA molecules which will lead in the end to protein. And these regulatory regions are usually characterized by binding of so-called transcription factors. So these are proteins which can bind sequence specific to the DNA and then control the expression of nearby genes or also far away by the so-called process looping. So, but I don't want to go into the three-dimensional structure, just take it linear, on a linear way, so we can isolate these regulatory elements by a technique which is called DNA-seq. And DNase is a restriction enzyme, or in other words, in human words, saying it's more or less like a scissor which is cutting DNA. And it's a very human enzyme, I would say, because it's very lazy. It's usually only cutting DNA which is highly accessible to DNase to begin with. And to begin with means in terms of 3 minutes. After 3 minutes, it's getting really also cutting a lot of other DNA. So we take actually advantage of this preference in cutting because regulatory regions where transcription factors are sitting are devoid of these histones, what I told earlier. So it's the DNA is very loosely, the proteins can interact with the DNA, and at the same time DNase can cut the DNA. And what we then do afterwards when we treated the cells with DNase, we separate these small DNA fragments from the bigger chunks of DNA and again subject them to sequencing. There we have also some nice people working in our group which are involved in getting the samples ready to sequence and do all the sequencing stuff. So it's very nice that we have this in-house. And what you then usually get is a lot of data. So you get like 20 million sequencing, what we call sequencing reads. So we have maybe 50 or 150 base pairs, 20 million of them, and what you of course have to do is to look where they're coming from. So you try to assemble them to, in this case, the human reference genome, and if you are lucky, it looks in a DNA experiment looks like this. that at a certain regulatory regions you pile up a lot of sequencing reads, meaning that you have isolate specific these regions, whereas in the case of the experiment would have failed, they would just be randomly distributed and you don't really have an enrichment of these open, so-called open sites. This is what it looks like when we combine RNA-seq and DNA-seq in the so-called genome browser. So that's a very nice tool to visualize genomic data. We have the human chromosome 1 located here, and we're looking just at a very small region down here, which is expanded. So we are maybe looking at 400 kb, so kilobases of genetic information, when there are some genes are located. And based on the RNA-seq, you might appreciate that this gene might be expressed, this gene is expressed, and this gene is also expressed, this one as well, but this one not so much, and this one also not so much. So it gives us a lot about information about which genes are active. And with the DNAs, we can now uncover a lot of sites in the genome which might be actually not overlapping with coding regions, or which might be within the gene, which are potentially to control the expression of the genes. And these are the regions we are highly interested in looking at. These are now data from osteoblasts and adipocyte differentiation. So we're getting back to our model system. And we are focusing here on genes which we previously defined to be upregulated during osteoblast differentiation on the left side. So these are genes like periostin or osteomodulin. So just from the name osteo, you might appreciate that they have to do something with bone. Whereas on the right side we have genes which are upregulated during adipogenesis, fatty acid synthetase, lipoprotein lipase, fatty acid binding protein 4, also something they have to do with adipocytes. And when we then look at the chromatin structure at these genes like periostin, here we have the differentiated stage, and here we have the undifferentiated stage. And you might appreciate that these profiles look very alike, also for osteomodulin and this IGF binding protein 3. Whereas when we look at these genes which have been related to adipocyte differentiation, the profiles look different. Then we have these peaks only in adipocytes, but not really present in the stem cells here for these 3 examples. So indicating that chromatin structure is different during adipocyte differentiation, not so much during osteoblast differentiation. We can also quantify and ask how accessible is the chromatin in the vicinity of these genes which are upregulated during osteoblast differentiation. And there we can see it's more accessible than close to the genes which are upregulated during adipocyte differentiation. So again, we're just looking at the stem cells, and it's a very nice confirmation that these genes which were already high expressed in the stem cells have also a more accessible chromatin landscape to begin with compared to the genes which are upregulated during adipocyte differentiation. So which indicates that enhancers near osteoblast gene seems to be pre-established at the stem cell state. And when you look more globally what is going on in the chromatin landscape using a Pearson correlation, so just asking how similar are these DNA-seq profiles compared to the mesenchymal stem cells, you might appreciate that we have a lot of red colors going here throughout osteoblast differentiation, meaning they are much alike. Whereas when we go throughout adipocyte differentiation, the color is getting colder, meaning actually that the DNA-seq profiles are very different between the samples. Which indicates us that adipogenesis is associated with more dramatic changes in chromatin structure compared with osteogenesis, which also reflects what we see on the gene expression level. So we came up here with a kind of a model system, meaning that when we look at enhancers or genes which are activated during osteoblast differentiation, it seems to be that the chromatin structure in the stem cell state is already open. There might be already a transcription factor sitting there. We can measure this by the DNA-seq. And these genes get activated during osteoblast differentiation, whereas the ones which are activated during adipocyte differentiation, shown here, they have a close chromatin conformation. The genes are not expressed in the stem cells, and then chromatin gets remodeled, so transcription factors are coming in, open the chromatin. We can measure this by DNA-1-seq, and then genes get activated. Of course, this is just a model and just very descriptive. What is going on during differentiation does not really tell you which factors are involved. But in order to identify the factors, we have the information about it, because we know the sequences actually at these regions. And as I said, transcription factors bind sequence specifically. We can somehow, based on the analysis of these sequences, get an indirect information about which transcription factors might bind in there. And then we have a guy in our laboratory called Jesper. So he actually developed a pipeline to use a lot of our genomic data to get a potential so-called key regulators, so factors which might be involved in differences between the samples. And I have to say that he's not just made this tool, he's actually really a driver of a lot of the stuff which we have going on in our lab. So he develops a lot of tools and he's very, very experienced and skilled in bioinformatics. With respect to the tool he developed here, was that he was using a mathematical model to evaluate the activity of a certain transcription factor based on the DNA sequences we found in these so-called regulatory regions. So how does it work? Or what is the underlying mathematical model? So the idea is a certain kind of activity at a certain regulatory region which we can measure experimentally in the laboratory, like DNA-seq or doing a chip-seq, for example, against a certain factor. It's just an additive function of all the factors or all the motifs. So these are the sequences which are associated with a transcription factor, which are found at a certain site. So indicating here that the activity of enhancer 1 is just 2 times the green motif, 1 times the yellow, and 1 times the purple. So it's a very linear model. And we have about 20 to 15,000 enhancers, and then we can do kind of a rich regression to actually estimate the activity of the motif. So the only thing we don't know is this one, but we know how many motifs we have. This we can figure out by analyzing the sequence information. This is something we can measure in the laboratory. And then we can estimate which motifs might be most contributing to a certain kind of activity. Here we have all the motifs, so reflecting transcription factors which are changing during differentiation. In our database we have maybe 1,200 transcription factors we could estimate by this approach. And here in our system we have 500 of them which are changing their activity during differentiation. And what I just want to highlight is with our approach we find a lot of them which are known to be involved in osteoblast and adipocyte differentiation just as a control. So all the red ones are key factors for adipocytes, the blue ones are key factors for osteoblasts. We find them. That's nice. But what we also figured out is when we're looking just at this heat map, how motif activity develops, is that we have a very, very big group down here, which seem to be active in the stem cells, indicating by a red color, but just decreasing throughout adipocyte differentiation, and they maintain during osteoblast differentiation. So these might be factors which are already present in the stem cell, and they might already shape this pre-establishment of regulatory regions near osteoblast genes. If they do so, we had to go back to the laboratory, and actually the way we are testing these is just by removing the transcription factor from our stem cells, and then let's see how they are differentiating. So we selected 12 transcription factors out of this big group, out of 200 here, knocked them individually down, so this is what we call it in the laboratory, so we remove it from the stem cells. And what we then measure is alkaline phosphatase activity as a readout for osteoblast differentiation. So this is highly present in if you stimulate the cells to become osteoblasts. Here on the left side we have our controls. These are non-differentiated cells. You can see that alkaline phosphatase activity increases during differentiation. And then if you go then to these different kind of samples, here are all the ones where we remove one of the transcription factors. And you might appreciate that a lot of them actually had a bad influence on, or removing them has a bad influence on osteoblast differentiation. While at the same time, when we measure lipid content, so how much lipid is formed during adipocyte differentiation, then we have actually an increased lipid content compared to the controls when we remove the factors. So some are really indicating that they're sitting at the edge. You need them for proper osteoblast differentiation, and actually they seem to prevent adipocyte differentiation at the same time. The nice thing working with genome-wide data is that you don't have to generate so much data yourself. You can actually download a lot of stuff. And we did this in this case for these factors which we locked down, because we were interested if these also play, if there's any kind of a hint that they have a physiological role in humans. I mean, it's very hard to do a knockdown experiment on a human. We don't get so many subjects to do these kind of researches. But you can do more like correlative analysis. So what we did is we looked into published data where people actually looked at mesenchymal stem cells from the bone from patients which have osteoporosis and patients which don't have osteoporosis. And highlighting here we have the transcription factors we knocked down, and what this heat map just shows, so as soon as the color gets purple means that this factor is differentially regulated between people having osteoporosis and non-osteoporosis. And then we just ask if if this is just true by chance. So we ask how much is the chance that the transcription factor by random is regulated by the process. And we find here a nice enrichment of our factors of interest, so they are more likely to be regulated during this kind of process than a random transcription factor, or all in this case. And we did this for a lot of data sets out there, so we not just look at osteoporosis, we also look at aging, we look at body mass index, obesity, insulin resistance, there are various conditions. And the take-home message is here is that these factors are often more likely to be regulated during these processes compared to a random transcription factor. So also indicating that they might play a physiological role. This leaves us in the end with a model system where we actually say that we found a transcription factor network, what we call it, which is present in the mesenchymal stem cell. And they actually already act on the chromatin, and it's very important for activation of osteoblast genes, while at the same time it seems to be that they have a repressive function on adipocyte differentiation. Bringing this into more physiological context, what does this mean? So our interpretation actually is that these kinds of factors are very important to rapidly induce osteoblast function, because in the case of a fracture you would immediately want to go that bone is remodeled and bone is reconstituted. Whereas adipocyte differentiation, so the expansion of adipose tissue, does not need to be a rapid process, because this is actually something you would like to induce if you know that you have continuous nutrient supply, and not just by just eating one burger today that you immediately increase adipose tissue at the same time. So asking if fat cell is the easy choice, naturally not. It's actually a tremendous effort the cell has to take to become an adipocyte, especially compared to an osteoblast. But you could imagine that diseases like osteoporosis, aging, body mass index, they actually have an effect of our transcription network, which enables our cells to rapidly become osteoblasts. If you disturb this network, you might remove the barrier, and then it's just during aging it's more easily to become a fat cell, and that's why you might have infiltration or even de novo adipogenesis going on during aging. Does this help us in the clinic? It might be, because actually enhancing osteoblast function is something which is not really present in the clinic. So instead of just saying that the X-ray showed a broken rib and we fix it with Photoshop, it would be actually nice to use the knowledge we have to actively drive osteoblast function. So far what is taking place in the clinic is only preventing more bone loss. So you get inhibitors for the Osteoclast, which remove the bone, but there are not really any agents which increase osteoblast function. So this is really an interesting research field to go on. And by this I want to end, and also have to say thank you to a lot of people, especially the ones which are involved in this project, like Susanne heading the project, and then we have Anders, Jesper, Mette, Martin and Elvira, who were heavily involved in doing experiments and in the data analysis. Collaborators like Mustafa, Jan Baumbach and Camilla Scheele, who provided bioinformatics skills but also access to the primary samples. And then we also have a lot of people in our MSc differentiation group which were not directly related to this project but now working on projects which have erased from all the data sets we have generated. And of course our sequencing guys who prepared all the samples. Yes, and thank you for your attention.
Thank you very much, Alex. I hope you have lots of questions.
So let me start by asking, so does it mean that the osteoblast differentiation is more dynamic? So basically, so let me put it the other way. So because you need to have a breakage of your bone, right, in a normal process to activate that differentiation as opposed to an adipocyte, does it mean that that process is more dynamic compared to an adipocyte differentiation process, which is less dynamic compared to osteoblast differentiation?
What do you mean with dynamic in the way of it, how a gene expression is coordinated?
Yes.
So I think it's more streamlined during osteoblast differentiation. And I think especially as there's only, it seems to be a very small modulation of these factors driving osteoblast differentiation, it might be actually something which is much more related to the metabolic state than like adipocyte differentiation. So it seems to be, it's easier to influence actually compared to adipocyte differentiation. But in terms of dynamics, it's hard to say. I mean, it's very rapid, osteoblast function. And mesenchymal stem cells, they produce, as I said already, a lot of these collagens and alkaline phosphatase. They produce it at the baseline. So it's just like giving them a boost. And this seems to be very streamlined, so very coordinated at the same time. However, it seems to also be very dependent on just a single factor. So it's not cooperative in a way that you can say it does not matter if you have one or the other signal, it will still continue. You still need input from a lot of sites, but it seems to be very streamlined. Just from the data what we have. Yeah.
So it seems to be a lot of trial and errors, of course, it's experimental testing. But sometimes it's also nice to have the theory we need, because if you want to, make some trial on humans, right? It would be nice to have a theory that tells you're not going to kill if you produce more of that or less than the other stuff. Is there a kind of a theory developing behind this, beyond these results? I mean, what can you actually take home as a message which is sufficient in general? And there is a second question is that there's a lot of machine learning that allows you actually to really, in principle, through artificial intelligence, which is technically not done on machine learning, you can actually have a better visualization of the data, which can fool an eye, but it cannot fool actually machine learning. Right? So how do you see these two things? One is how do you make a theory out of it, which is allow you to actually predict rather than caustic. And the other one, how can you use modern methods like machine learning to actually make a more sense of all the data you have?
So let me just start with the second. So what we included in this project is actually a lot of machine learning, because we anticipate factors just based on sequences we find in the DNA. We don't know that the factors are sitting there. We just predict it. And we have this model which just tries to model what kind of factors are important. It's in a way also machine learning that we can challenge the system. So what we also do is a lot of what's called leave one out analysis. So we're removing a motive and actually see if we get an error, which also indicates that the factor might be even more important if we get an error in the model. So we use these kind of approaches here. With respect to the first question, that's a good thing to actually challenge our hypothesis. What we would like, or what we are going to, is we really would like to see if this is really happened in vivo, in the human subject. Of course, we do not manipulate in that way, but what we have now access through the Odense University Hospital is that we can call people. So we got access from the ethics committee to take bone marrow biopsy and do the process which is called single cell sequencing. So we really want to look if what we measure on bulk, right, which we measure on a population of 1 million cells, is this is happening at the same time in one cell, or actually do we have different kind of populations to challenge our hypothesis we have? And in the long run, of course, it would be nice to generate agents, but I guess this is not really the first place of our basic research here in the gene transcription laboratory.
Yeah, so I was sort of mesmerized by this 3D plot which you showed, that sort of, I mean, irrespective of the sort of origin, where because I mean they always follow the same trajectory if you give them these cocktails in vitro. So I mean, do you think this is partly an in vitro phenomenon, that you basically expose them to the same medium, the same cocktail, so you sort of artificially drive a certain trajectory which maybe not would be there in vivo that much? And maybe the other question is, so what are these differences? So I mean, why is the trajectory, the same, but the underlying differences are actually huge? And how do you interpret them? Is the cellular context in their, let's say, organ of origin, or?
So one needs to say that in this case, principal component analysis is a very nice tool for visualization. But you also should keep in mind what kind of data you feed into it and what you get out. So it represents you more or less the biggest variance it finds in the data set by its components. And of course you could just imagine, okay, these cells, what is going on during osteoblast and adipocyte differentiation is very similar. You would think of it in a biological way, but you could also say that this is the biggest variance, is that the similar response to the cocktail. So actually what it does not really capture is how osteoblasts differently respond to the cocktail or the bone marrow derived form compared to the adipocyte derived form. This is something you don't capture. It is known from in vivo experiments or transplantation experiments, only, at least from mice, I can only say from mice, only the stem cells which were isolated from the bone marrow form bone in a transplantation experiment, not the others. If this is something related to differentiation, or if this is something related to which is already present at the cell to begin with, I don't know. This is also a question which Mustafa from the bone group actually also asked me quite heavily. It's always the same question: why do these cells not form bone in vivo, the other ones? And the simplest answer is that it might be something which is totally different from begin with into stem cell. It might be not something which has to do with differentiation. It's just that they have a certain kind of probability or not. And this is something people never looked at, really, the tissue origin in this case.
On the machine learning, your model where you also briefly mentioned you use rich regression. Do you these nonlinear portions where you have an interaction, do you identify those through rich, or are they pre-identified, and do you use the rich to then instead assess which are the most important, or both maybe, which is or could you elaborate a little bit on how you use the rich there?
Also, so how it's technically done, or from the mathematical point of view, I really have to say that I have to really say I don't know. And I think also even Jesper, who implemented it, also is not very keen on the mathematics. It's something, a model which has been already published. We applied this, we expanded this, we put more motives into the database, so we take more the biological use out of it. What the model tells you, what is also something which I didn't mention, is we get these kind of activities of all the factors. And the sum of the activity is always 0.
Yeah.
So we can only just say this is more a little bit like the others, but what it means in absolute terms, we don't really know. So it's just the sum is 0, and we're looking at changes between the samples. And these changes, especially if you also do these modeling and rich regression, even if you say you have the motive on the very top, or let's say it's on place 10 in one samples and then it's on place 20 in the other, actually does not necessarily mean that the motive is less active, but it could be just that 10 others are more active, right? It just goes down in the model. So this is also a kind of tricky thing. But we are more keen on, if we do these kind of leave-out analysis, that we really get a big arrow. If we don't have this motive in the model system, we cannot really solve the equations nicely. And this is, I think, a nice indication.
What is your benchmark? I mean, it's all relative. So what is your overall benchmark? I understand you measure it within a sequence. You mentioned the activity with respect to each other. First of all, this activity doesn't have to be linear. That is an immense oversimplification.
It is simplification. There is always a simplification.
But
if you use
there should be some
model.
kind of always a normalization to in actual known activity where you know it's linear.
So
That's I wouldn't trust it. Right?
Speaking about a biological system, there's a lot of publications out there which actually say that these transcription factors are not just working in an additive manner. So they are acting cooperatively. The problem is that but the problem is still, compared to the mathematical model, is that you usually just investigate, like you have 2 factors you're focusing on, and then you just see if you have one, there's a little bit going on, if you have the other one, is a little bit going on, if you have both, it's a lot,
Yeah.
biologically.
So, but
The
I assume
problem-
also, sorry to interrupt you, because the way to measure this is with the correlation matrix. I assume you also measure the correlation matrix, right? The correlation matrix tells you how many 2 different factors, interact with each other. And if the correlation is very big, then the 2 are not really linearly adding to each other.
Yeah.
So there is a correlation matrix to calculate.
But I would just say the problem in our biological systems is that we always just focusing on the 2, right? We actually don't know if you have put both together, if the edit or the, what we call it, the cooperative effect is really due to the effect that you have cooperativity, or it's just due that if you put both these 2, you get 500 others as well, and it could be still just additive things.
But even that,
we don't know.
Even if a 2 by 2, even with 2 single elements, right? You can still calculate a correlation matrix, how much one influences the other. You can actually calculate that from your data set. Because when you do your statistics, besides calculating the mean square, you also calculate automatically that you can calculate the correlation matrix. So that tells you how much each factor influences the other.
Yeah.
This is what you
You need the data for that.
don't you already have it from the minimization? So otherwise you wouldn't even know how to get to the linear term.
Yeah. So
That's what I learned in school.
no, but the way we benchmark the model is that we use published data sets. So what we can predict from our model is that this factor is important at that side, that side, that side, that side, due to the error we get out. What we then ask, there are published data sets, which is it's called a strategy, it's called ChIP-seq. So you pull out this transcription factor with an antibody and just look what kind of DNA is sitting there. You sequence it, and then you can then do a correlation matrix. And then we can see that our model is better than predicting sites than random, better than just using the motif itself. And we can also benchmark it, and that's actually the coolest thing, if you would remove the transcription factor from your system and just measure with the effects. Our model is actually better in predicting the effects than the technique where you isolate the factor with the antibody. So, which also indicates that's a problem with biological data, there's a lot of noise. If we look at our transcription factors, some of them bind at 100,000 sites in the genome. How can you make sense of it? Maybe in the end, if you remove it, it's only 100 genes which are affected. Still it binds 90,000 sites. So that's very tough from biological data to make means out of it, and actually the modeling is better at doing it. So that's quite fun.
Can I ask a non-mathematical question?
Yeah, it would be nice.
So I was really, I mean, if some what you basically said is that, you know, that there's basically a roadblock in place which basically prevents that these MSCs skew automatically towards fat cells, right? Which makes a lot of sense. But in the same time, you said that actually during aging there's this fatty degeneration. And I mean, of course, all the transcription factors which you called, I mean, in a way are a proxy for physiological changes, right? You have start transcription factors, I mean, it's growth hormones, type 2 cytokines. I mean, so if you look at the transcription factors which you identified and their alterations during aging, does that tell you something about the underlying physiological changes?
We have not looked at this at all. I mean, what we only looked at is that some of these factors are differentially regulated in stem cells from osteoporotic or old and young subjects. This we know, more than by chance. If they are implicated, I mean, and the problem is with these kind of analysis, you never know the hen and egg problem, right? Are these genes just regulated because due to the aging effect and now the cell actually would counteract this one? Or is it actually the causing effect? So that's why it's really tough to make a clear cut. What we would like to do, what we haven't done so far, but we have the data, is actually to look if the sites where the motifs are, so these factors, if they are enriched for disease-associated variances, which have to do with bone mineral density, which have to do with BMI, something like this. That would be cool. We have the data, but I haven't had the time to look at this.
Yeah, I mean that's one way, but I mean, let's say the physiological regulation of transcription factors is probably way stronger than, let's say, genetic variants at a certain site. Right? So.
But some of these factors are actually not changing expression, it's just activity. So some are also changing expression. So the ones we looked at, maybe half of them are also regulated on the transcriptional level, so meaning the activity decreases because the factor is not there anymore. But some are staying constant, meaning it's post-transcription or post-translation.
So maybe one final comment. So you have this group of 200 transcription factors that show pretty similar change in their activity profile during both differentiation pathways. And you would think there was a lot of redundancy between this whole group of 200 transcription factors, yet you see that if you just remove one of them or knock down one of them, there's a dramatic effect on the ability to induce both differentiation pathways.
So that means the correlation matrix 100%.
Yeah. So
it has implicate.
there's something, I know something we discussed, but maybe you want to elaborate a little bit on that.
That is exactly the demonstration that the correlation is basically huge. Between different factors.
Yeah, so I mean that's a good question which you not actually would anticipate it. So you actually would rather go for the other that is more like a safe pathway, and if you remove one or two, it's not really that important because the cell has other possibilities to circulate. Actually, it seems to be that it has not. This might be actually also due to the effect that so what we know is that these factors are also sitting together quite often at the same place, so and control a lot of the same genes. So this we know. In what we don't really know, this is something that would be interesting to investigate. So how does actually chromatin landscape look like if we remove one factor? Does it actually affect all of these where the others are sitting, or how is compensation going on? So It's one of the toughest questions, and I hope the reviewers are not asking that.
Well, I think you did these network analysis.
I'm a bit skeptical of what you actually approach.
So where you think that, where you show that the factors is very highly connected, so you can, I think, you know, my interpretation would be, it's just predicting that if you pull out one factor in the big network, it affects all the others, one way or the other, whether it's by sharing sites or it's affecting the regulation of other factors.
Oh, sorry, I mean, I was thinking about, just a quick one on, you said leave one out. I thought, shouldn't you leave one out and all of that, then two out, then three out, then four, and you should do the entire thing. It's another way of saying the correlation metrics, by the way. So leaving one out is an indication, but surely not enough.
Oh, no, definitely, yeah. But I mean, the algorithm which we have right now is still takes 1 day to run with the data set we have by just leaving one out.
Okay.
If you leave two out, you have to do it on all combinations of.
Exactly.
Right. So
that's the point. That's the
so
point.
computing power is getting.
No, but you can.
Yeah.
So just a minor point, but so let's say you have 10 factors that you're evaluating the importance in a given system. What you can also do is when you're calculating the ridge analysis, you can also rank them based on their p-values and do an iterative process with every leave one out. And what that gives you is a rank measure to say, this particular guy exerts most importance, right? So when you knock that candidate out, you should see the massive phenotype. And so it's just a ranking analysis that allows you to get some confidence measure into doing that. And to address some of these questions.
It's a good point, but you also, when I need to comment on that, because what we do is actually we combine more or less all the time points we have. So meaning that actually, I mean, you can do it on a single time point, right? To rank each factor at a single time point. Our analysis is based on the whole data set. So even if we have this factor, we don't know if this factor is important at osteoblasts late in the stem cells and the adipocytes. We don't have this information because the model is getting better if you just combine all the time points. You can do it on single time points, but then the prediction is worse for the single time point. So, I mean...
But that's your probability for, so this is a combined effect for different processes, so that too there is a statistical way to take that into account, right? So that's not just statistics. So technically you have the product of each thing happening at each time, and then the product of the probability is the final probability. You measure in the final one. It's not that you don't have that information. So I would say that is, statistics takes into account that many things. It's combined probability for independent events. That's actually what you do. But I mean, I appreciate the effort. So I'm saying that I think it was asked about it because it is interesting.
I think we should thank Alex once more.